right, we're going to talk about the process of crossing over. Crossing over also happens in the meiosis process, and it's going to create quite a bit of diversity among cells. And we'll talk a little bit more about that later. But we're going to go through each part separately, but I'm scanning out like this so you could stop it and take notes. Okay, so we're going to start with the first thing, which is the one homologous set. So like I said before, in humans we would have 23 sets of these. I've just indicated one of the chromosomes is colored dark just to show that it's different, because remember that each of the homologous sets of chromosomes, the homologous sets of chromosomes have genes on them that are the same for the same traits, but those traits may actually have different alleles, or they may be different versions of each of those genes. And because each chromosome has hundreds of genes on them, it's unlikely that the homologous set of chromosomes, that one chromosome would be exactly like the other one. In other words, the mom's chromosome of one set is not exactly like the dad's because of the different allele possibilities. Okay, so in this case, I just have, one homologous set. I've just drawn dad's as dark and I've labeled it capital A and little a just to kind of keep track. Okay, so this is the beginning of the process. If we were doing a cell division process, it would be in G1. So I'm just talking basically about what's going to be happening in meiosis. Meiosis has two cell divisions. Basically, all of the stages that we talked about before will be gone through twice. So that is, we talked about prophase, metaphase, anaphase, telophase, but in the first time it's called those 1, and the second time it's called 2. So I'll write that out for you in a little bit, but right now just to kind of generalize. Okay, so the next thing that would happen is replication. So replication, when that happens, that each of those homologous sets is going to replicate itself. In other words, it's going to get a sister chromatid, and remember that in replication, that the sister chromatids are exactly alike. So in other words, capital A and capital A prime are going to be exactly alike, and same thing for little a and little a prime. So I've drawn them just uncolored and colored just to kind of represent that. Of course, this would be happening during the synthesis phase of interphase 1. Okay, the next thing that happens, and I've kind of indicated that we've gone through several phases here. I forgot to do it the next time, but anyway, in prophase 1, what is going to happen is the chromosomes will wrap together. And when they wrap together, what's going to happen is they will join up, and proteins and so forth sort of help them intertwine. And every place that they touch, they are going to exchange information. So in this case, I've drawn, so they're going to exchange information during prophase 1. This is the process actually of crossing over. In other words, crossing over happens in prophase 1, and it is doing the exchange right here. Now those joinings are called chiasmata, and they during this phase, this structure is called a tetrad. We'll talk about that again in a little bit. Okay, so as they exchange information, the next one here is showing you they would exchange information, and as they got pulled apart in anaphase 1 and ended up in separate cells in telophase 1, they would look like this. In other words, the first one, I forgot to label these, whoops. The first one over here, oops, sorry about all the messes. This is capital A, and this would be capital A prime, and this would be little a right there. And right here, this would be capital A prime, but this would be little a, and this would be little a prime over there. All right, so you can kind of see how they basically, little a and A prime have exchanged pieces. Capital A and little a prime have not in this picture. Most of the time, though, in reality, all of the chromosomes would have had some exchange. All right, so now through the process of meiosis, what will happen through the next phases is that at the very end, I'm going to end up with cells that have one of each of those homologous chromosomes, like we were talking about earlier. These are the haploid cells. But notice that the chromosomes are no longer like they were at the beginning. In other words, I don't have two clear ones and two dark ones like you might expect, right? Two clear, two dark. I instead have one clear, one exchanged, the second exchanged, and one dark. So in all four of the cells created, each homolog or each part of that homologous set are going to be different. So this whole process is called crossing over, and it creates quite a bit of diversity because remember, this is happening to all 23 sets. Okay, now this is just an overhead. This is in your textbook of the same thing, and maybe just slightly better drawn than mine. So check out the overhead, check out the figure in your book.